This video demonstrates a procedure for determining cell viability in cells expressing GFP using the Tali Image-Based Cytometer. GFP transduced cells are stained using the Tali Viability Kit, Dead Cell Red, which stains all of the dead cells red with propydium iodide. The cells are pipetted into a Tali cellular analysis slide and loaded into the cytometer, which acquires and analyzes bright field and red and green fluorescence images. Histograms are then generated to display cell count, cell size, PI fluorescence intensity, and GFP fluorescence intensity. When compared to traditional flow cytometry, Tali can provide similar data regarding cell viability and expression. However, this image-based cytometer provides additional visual information about the cell population being examined.
The main advantage of this technique over existing methods like flow cytometry or fluorescence microscopy is that the Tali image-based cytometer provides both quantitative and visual information simultaneously about your cell sample. In addition, the Tali instrument is easier to use, is less expensive, and has a smaller footprint than either a flow cytometer or a fluorescence microscope. This allows researchers to quickly perform quantitative assays like viability in GFP-expressing cells on the benchtop.
In this procedure, U2OS cells at a concentration of 1 × 10⁶ cells per milliliter were transduced with nuclear-targeted cell-lyte GFP viral construct. To stain dead cells with propydium iodide, transfer 100 microliters of the cell suspension to a new microcentrifuge tube. Note that a concentration of 1 × 10⁵ to 1 × 10⁷ cells per milliliter is recommended for the assay, though the concentration does not have to be exact. Next, add 1 microliter of Tali Dead Cell Red reagent.